If you go cast your minds back to, say, 2015, 16, 17, something like that, state of knowledge about nematodes in vineyards in BC was largely based on samples that had kind of gone in on an ad hoc basis to the plant health lab, and where they'd been doing diagnostics, and then also a smattering of samples that I had been doing off the side of my desk, working with various vineyards, et cetera, and so on. And so at the time, we knew that there were several broad groups of plant-parasitic nematodes present in BC vineyards, starting with root lesion nematode, which is the one that was probably most on people's minds at the time. Tree fruit production. This nematode is very important in tree fruit production. So for decades, we've known about its importance in tree fruit production. And there's also a Pratolynchus species that is a well-known pest of grapevines. And so this one was obviously on people's minds. Dagger nematode, people know vector Nepoviruses. And so that group of nematodes was on people's minds. Root knot nematodes, if you're online Googling nematodes and grapes, most of the information you're going to get revolves around root knot nematodes. So those were on people's minds, and we knew that they were present. Then ring nematode was an interesting one, not very prevalent back in those days as far as we understood, but I happen to know from work that I'd done and been involved in Oregon in the late 1990s, early 2000s, that ring nematode can be a significant parasite. It's not, in most grape growing regions, it's not the highest priority pest, but it can be a significant parasite depending on its abundance and things like that. And because of that, in the sort of around 2010, I actually did some what we call controlled inoculation studies, which I'll explain later, to demonstrate that a BC population of ring nematode could in fact reduce growth of grapevines. So even though we didn't know the full extent of it, though, at that time. So again, the prevalence of these groups of nematodes were not well known at the time, and but likewise, the potential impacts weren't really completely clear in terms of the exact amount of damage that we might be observing across the industry at that time, in part because we didn't know their prevalence, but also because species by species basis, the actual abundance or the population densities of the nematodes is a determinant of their potential damage that they're having. And so also then that behooves us, if we're starting to look at the distribution of nematodes and which ones we have in the Okanagan Valley, to keep track of how population densities are affected by things like rootstocks and soil properties and soil and water management practices. So that's kind of the background or context when we started this project. So we started right off the bat in 2018 with a more systematic survey rather than just kind of cobbling together information from ad hoc samples that were submitted to the Plant Health Lab. And so we sampled 62 vineyards in the summer of 2018. And one of the first things we learned, the top key finding, was that those ring nematodes I just talked about were really widespread. 82% of vineyards that we sampled had ring nematodes. They ranged in abundance from 0 per 100 cubic centimeters of soil or 100 milliliters of soil, which is a standard sample size. They ranged in abundance from 0 to upwards of 800 per 100 cubic centimeters of soil, which we know from some of that previous work in Oregon and other places are damaging levels of populations, for sure. And so we think what's going on here is 20, 30, 40 years ago, as the industry was really growing, a lot of sites were being planted, a lot of vineyards were being planted into old apple sites, in some cases old cherry sites, but mostly old apple sites, or perhaps previously uncultivated soil or maybe forage production land. And so at the time, these nematodes were not really on people's radar. Then and another reason that we think that they weren't really on the radar and they didn't seem to be very prevalent based on samples that had been analyzed previously is that the extraction method required to recover this particular group of nematodes is different from the extraction method that most diagnostic labs use. So we'd probably have been overlooking it to some degree, but to some degree it just wasn't very widespread back in the day. Now it's a different situation. We've had 30 to 40 years in some of these vineyards for these residual populations to build up through time, which is the way nematodes work. So in addition to the ring nematodes, dagger nematodes were relatively widespread also, 77% of sites. This wasn't as much of a surprise. There was a systematic survey done in the mid to late 1980s, completely different vineyards back in those days, but we knew that it was fairly widespread. And I'll talk about species of Xiphinema later. Root knot nematodes were not widespread. 25% of sites, and we know that all the populations in the Okanagan Valley, at least, are Meloidegon hapla. It's known as the northern root knot nematode. Among all the root knot species of root knot nematode that you might read about impacting grapevines, it is the least damaging on a sort of nematode to nematode basis than the other species that are found in California, for example. So that's just a quick overview of what we found in the Okanagan survey. And this has been written up in a paper that if anybody wants to look at it in more detail and et cetera and so on. Nova Scotia, I had the great opportunity to team up with Deb and Harrison, who sent me samples from approximately 60 vineyards in Nova Scotia. And we were able to take a look at the nematode populations they had there. And this is kind of the first foray into nematodes in Nova Scotia at all. And not too surprisingly, after looking at the Okanagan data, we found ring nematodes in about 80% of their vineyards as well. In their case, we did find at least one other species than Mesocrichinema xenoplex, which is the main species that we find in BC and also the main one that causes problems in vineyards around the world. So they may have more of an interesting mix there. Dagger nematodes were also relatively prevalent, 57% of sites. And I'll back up, and I put the text there in red about dagger nematodes, and I had that there before on the BC one. So the presence of these nematodes is, they're not on their own, they're not all that damaging, but it's highly significant if we ever get Nepoviruses into any of our vineyards, because these nematodes as a group vector Nepoviruses. And there are some specific Nepoviruses that go with specific nematodes that I can talk about later. Again, just to be brief, getting through this, root knot nematodes were more prevalent in Nova Scotia vineyards than in BC. And again, just as an initial foray, we couldn't go into much more detail than that, but it's definitely something that deserves follow-up in the Nova Scotia industry. And again, we've written up this in a paper that people can access if they want to follow up on it in more detail. Bottom right-hand graph is just simply relationship between vineyard age and ring nematode population densities. Again, just kind of confirming what we observe in general with newer industries. It takes 10, 20, 30 years for populations to build up in vineyards from an initial introduction. So that's just consistent with that general trend. So some of the more experimental work that we did as part of the project from 2018 through 2023 had the opportunity to team up with Tanya Vogel, who's basically our specialist in crown gall disease, and Louise Nelson, bacteriologist at the time at UBC Okanagan, and graduate student with Tanya and Louise, Portia McGonigal, and to take part in a study that basically Tanya was leading, but it was a study to look at how compost amendments applied to a vineyard with crown gall issues might influence the spread or further incidence of crown gall, but also how that might relate to changes in nematode and plant-parasitic nematode populations. There are some underlying hypotheses there that we were interested in pursuing, but we're looking to see if there were field-level observations or data on responses of populations that would lead us, it would support those basic hypotheses. So key findings from this study. So we ended up sampling. So in this test vineyard, lower left-hand photograph, the vineyard was about 4 years old at the time that we set up the study. We had 3 different composts that were applied at what I would call agronomic rates, relatively low rates, surface applied, so not applied as a really high rate amendment. And so we had those, and then a control where the compost wasn't applied. We monitored nematode populations 2 times per year over 4 years. And then likewise, Tanya, Louise, and Portia did lots of careful analyses of crown gall at the site and the spread of galls and things like that. So key findings, ring nematode populations remained low at that particular site, which is really strange throughout the entire study. And we don't understand what was going on there, but it was a low population to start because it's a fairly young vineyard and it had been planted into what was previously a pasture. Dagger and pin nematode populations increased throughout the 4 years of the study. The bottom right-hand graph shows the increase and different among different treatments in the dagger nematode populations, but there were no effects of the compost amendments in the time frame of this experiment on either of these nematode populations And so there was no relationship with changes in crown gall either at this particular experiment. So just a quick overview of that. Portia is in the process of making final revisions on a manuscript on that work. I also got involved with Pat Bone and Carl Bogdanoff in an experiment they set up here at the Research Center to explore the influences of canopy-based, infrared temperature canopy-based irrigation scheduling that was set up in 2018. And so we commenced sampling in the various treatments and that, and we were really keen to learn how different irrigation practices that will influence then residual soil moisture regimes, how that might influence the buildup of the nematode populations, and if that relates then in turn to responses of the vines to the different irrigation practices. Unfortunately, partially due to COVID, the treatments were terminated in 2020. So before we really had time for the treatments to really kind of have a sustained influence on below ground biology. And then Pat and Carl retired, of course. And so we carried on sampling, and we attempted to do some statistical analyses using the data from all the plots to relate population densities of the nematodes to growth suppression. And the bottom left-hand graph is just simply a graph of the spatial distribution in the experimental plot area, with each point being one of the individual plots within the general area. The graph on the lower right is actually, I probably shouldn't even put this up here because it's going to distract people, but it's what it's showing is actually, it looks like an increase in trunk cross-sectional area in relation to increasing ring nematode populations, which is completely counter to what we know about the actual parasitic impact of these nematodes. But I think that this is, it just shows when you try to do this kind of work in blocks where there have already been treatments imposed before, and we're probably seeing confounding effect of plots where vines had been, their growth had been increased for other reasons, and there had been nutrient kinds of studies even before Pat and Carl in there. And we're just seeing a response of the nematode populations to whatever those treatments were. Quickly, I'm going to go on to the kind of the third pillar, if you will, of the work, or a broad objective of the work that we did. And it's under the category of what I called controlled inoculation experiments, where we take, develop pure populations of nematodes of interest, rearing them in the greenhouse, and then we can do controlled inoculation experiments to ascertain whether they're, how they're parasitizing the plants, and whether they have any observable effects on plant growth, at least under greenhouse or what we call microplot kinds of conditions. One of the most important areas of work that we did here was with root lesion nematodes, which I mentioned right off the bat, but I didn't mention in terms of the key findings. So it was really widespread also. And as I mentioned, these nematodes are well known as parasites of tree fruits, and we've often just kind of assumed that they're probably important in our system here. So I teamed up with Inga Zasada, colleague of mine down at USDA in Corvallis. She had populations of Pratylynchus neglectus and Pratylynchus penetrans. I had populations of Pratylynchus neglectus and Pratylynchus penetrans. We both put those onto self-rooted Merlot and Chardonnay, and we both repeated them, and we were not able to get those nematodes to parasitize those grapevines. So we can rule out Pratylynchus penetrans and Pratylynchus neglectus as parasites of grapevine in the Okanagan Valley and the Willamette Valley at least. And so what we think is happening is that due to past infestations, these nematodes all have relatively wide host ranges. They're just persisting in vineyards on cover crops, alley cover crops, weeds, what have you. And we're picking them up in soil samples, even from the vineyard root zone, but it's because they're straying around in there. They're not actually parasitizing the grapes, so they're not important. So it's important to remember as growers, when you do get nematode analyses and you see those numbers, Pratylynchus and some sort of large number, it's important to know you can rule that out. But another important thing though, one species of Pratylynchus, known as Pratylynchus vulnus, in Mediterranean regions, California, Mediterranean, etc., is a highly significant parasite. And so we need to stay vigilant for it, because although it's relatively restricted to Mediterranean regions, it is actually present as far north as Idaho in North America. And we think that it would probably be able to survive here if it arrived here. So we still need to stay vigilant, but it's really, but we have to do it on a species by species basis. And finally, ring nematode. my technician and I, we developed pure populations of it, and we were able to contribute in this cycle of funding to greenhouse and microplot experiments that Jared Rykan, who was a PhD student with Jose at the time, and we were able to contribute to some of his studies via our work with that nematode. And I hope Jose will talk a little bit about that. So just to wrap up, what we now know, we do not know, but we might want to know, or in other words, to summarize, or future directions. So we have a very different context right now than we did at the beginning of this project, in which we were just thinking, what is the role of nematodes in vineyards, existing vineyards, et cetera. Right now, we're talking about what are the role in replanted vineyards. So I think it's important to keep that in mind. It kind of highlights things a little bit differently. So again, I point to our findings on ring nematodes. And so I will just go out on a line and not talk like a scientist usually does. And I'll just say they will be affecting replanted vineyards, depending on the populations that are there. But we have lots of vineyards now that do have larger populations that we would not have had 20, 30 years ago. The extent of the damage, it's hard to say. These pests are chronic long-term pests. They're not going to just kill the vines immediately right off the bat. But it's important to keep in mind that they're going to be having an influence. Pre-plant damage thresholds, question came up a little bit earlier today. We really don't know what they are. People often want a pre-plant damage threshold. It's for a replanted vine, we can make some rough guesses. But once you get 2 or 3, 4 years down into a vineyard, to take a sample, get a nematode count, and then to try to attribute that to some sort of observation of vine growth is really, really difficult at that point in time. But that's an area that more work could be done. Interaction with Blackfoot and young vine decline disease, and I hope Jose might mention this as well. Root crown associated disease issues highly related to replant scenarios. We think it's highly likely that these nematodes may interact with the pathogens in these complexes. But experimentally, it hasn't really been done yet. So again, rootstock tolerance and resistance, clarifications on that. Self-rooted vines to ring nematodes, self-rooted vines are way more susceptible than any of the common rootstocks. None of the common rootstocks are resistant. And so people refer to nematode-resistant rootstocks. That refers to nematode populations, mostly root-knot nematode species in California or wherever they were doing that work. Ring nematodes, none of the rootstocks are resistant per se, more or less tolerant, but not resistant. And of course, learning more about how soil nutrient and water management practices might minimize buildup and investment in the future. Dagger nematodes, important to keep in mind, the species we have in British Columbia was identified a long time ago as a new species, Ziphinema bricalensis. It's a species within a species complex known as Ziphinema americanum. We know that they're vectors of Nepoviruses. One main Nepovirus, though, that is on most people's minds is Grapevine family virus. That one particular Nepovirus, however, is only vectored by Ziphinema index, which is a species that we do not have in British Columbia. So just to keep in mind. But we never have experimentally determined the Nepovirus vector capacity of Ziphinema bricalensis. Going back to Grapevine family virus, other Nepoviruses of concern, tomato ring spot virus and Arabis mosaic virus would be the first in mind. Tomato ring spot virus is largely considered to not be an issue in British Columbia nowadays, but perhaps some vigilance might be in order, as it seems to be occurring more frequently in Ontario and Quebec, is my understanding. And 1 minute, I already summarized about root lesion nematodes, highly widespread, but we can rule out Pratylingus penetrans and neglectus. So I think that's where a negative result is actually a good thing, but we do need to stay vigilant for Pratylingus vulnus. And I'll just point out, these are endoparasites, the root lesion nematodes, and so it's very easy for them to spread on nursery material. And I will stop there.